section with all of the speakers from this particular session. So please feel free to type your questions in the chat and we will relay those to the speakers at that point. For the remaining part of the section, we are going to be focused on the 4 talks that have been selected from the submitted abstracts. The first talk will be from Nicholas Cardenas from North Carolina State University on foot and mouth disease modeling in Brazil, the impact of a potential outbreak and control measures. Our second talk will be from Maria Victoria Iriarte Barbosa from Gininan, and she will be presenting main factors associated with FMDV introduction and infection spread during the high-risk period of the 2001 FMD epidemic in Uruguay. Our third speaker is Dr. Umanga Gunasekera from University of Minnesota on the use of slaughterhouses as sentinel points for genomic surveillance of subclinical FMD in Vietnam. And finally, we will close the session with a talk from Samia Metwali from FAO towards the secure elimination of FMD serotype C.
Hello everyone, my name is Nicholas Cardenas. I am a postdoc researcher at North Carolina State University, and I'm going to talk about the foot-and-mouth disease modeling in Brazil. So here the study area is Rio Grande do Sul, that is one of the biggest states located in the south of Brazil that shares a large border with Uruguay and also with Argentina. And one special thing of this state that is stopping the vaccination against foot-and-mouth disease in order to be declared free of the disease without vaccination. And this creates an urgent need to large scales preparedness for outbreaks and also for epidemics. So one thing that is important here in these populations is because they share a large connected network among the species. Here, the multiple populations are cattle, but also have a higher number of swine farms and a lower number of small ruminant farms. This is mainly sheep and goat. One thing important here are those farms are highly connected to the animal movements. So one disease is capable to infect other species through the animal movements, despite of the distance of these farms. So the means of this work will describe the foot-and-mouth disease spread potential using a susceptible exposed infected model that represent the dynamics at farm level and also at animal level. So the model simulation was stopped over 30 days in order to quantify the contribution of the animal movements and the distance proximity in the infection of the new animals. And also we calculate with the additions for the initial outbreak to the secondary cases. Okay, as material methods, we have here a large databases that we include cattle, swine, small ruminants, information regarding with movements to farm and farm to slaughterhouses. This is around 1.5 millions of records that comprises around 400,000 farms. That this is a large scale simulations, and we are considering around 20 millions of animals in these simulations. This is over 3 years of data from 2018 to 2020. So here are the how this models works. So we are simulating each farm. In each farm, we have 4 compartments that represent the status of health of those animals. So in each farm, we can find cattle, swine, or small ruminants, giving just one species of the 3 species. So in each farms, the animals could move from one compartment to other compartment according with transmission coefficients, and they are moved to susceptible to exposed animals, and then those exposed animals becoming infectious animals have the ability to infect another susceptible animals here, and then those animals could be recovered or not. So here we're modeling the interactions to cattle, swine to swine, small ruminants to small ruminants, but also we are modeling the other interactions among the factorial species, cattle to swine, cattle to small ruminants, etc. Here, I'm gonna talk about a little bit more about the models. So here, I'm representing the within farm dynamics. So I explained it before. This represents what is happening inside of each farm. But also we have records of the behaviors of the animals. So the animals, they are introduced into the susceptible compartments in each specific farm and each specific time according with the official records. But also we are considering the death of the animals. This is given for other disease or by accident. And also we are considering the routes of transmission. There are two routes that we are considering here. The first one is the animal movements. So according with the records that we have of the animal movements, we recreate a network contact. And in this, we are selecting randomly the animals that are gonna move from one farm to other farms. Also, we are considering the movement to slaughterhouses, subtracting those animals that are going to the slaughterhouses from the simulation. And finally, we are considering a kernel transmission where farms that are more close to the others, they are more likely to be infected than farms that are more far than the others. Okay, here we are considering 600 simulation where we put one infected animal at the beginning of simulation in one farm, and we are considering 30 days of simulations. Here, I don't have so much time to talk about the transmission coefficient, but we are considering transmission coefficient for most of the factorial interactions here. And also we are considering this transmission coefficient in this space. The result that we have here, in the y-axis, we have the number of the animals. And here we are representing the status of the compartments across the simulations. These results are the 30 days of simulation. So we can find here the bovines in general, they have more animals in all compartments. But also we can find here that the exposed animals at risk in the swine populations, they are a little bit higher compared to the cattle. But the number of exposed, infected, and recovered animals were similar between bovine and swine farms. On the other hand, as expected, given the lower number of farms and animals, we cannot find more than 20 infected small ruminants in this area across the all simulations. So we have here the same result, but this is grouped just for farms. So we can find here, we can get simulation with just one farm infected, but we have the worst case scenario where more than 40,000 farms exposed. But of those farms, we considering around the 100 for 1,400 infected farms. This is going to be the worst case scenario for these simulations at 30 days of simulations. Same results were extracted for the swine populations, and also for the small ruminant farms, we don't get more than 25 infected farms across the all simulations. So we're now going to explore what is the results across the space. So here, in this axis, we are considering the distance in kilometers from the initial infected farm to the secondary cases. So one farm infected here, we are measuring the distance to the secondary infected farms. And we are doing this for all the simulations for all the data. So the main conclusion here is in all the simulations, mostly all these infections keeps around the 1 kilometer to 20 kilometers. So for this plot, we could around 40 kilometers, but of course there are movements with more than 40 kilometers, but they are more likely to happen. And here we're gonna explain what is the contribution of the animal movements and the distance proximity in the infections of the new animals. So here is the summatory of new infected animals. And here we are counting this contribution among the days that we are simulating. So here we have the result for all the species, but we're going to split here. We can see here that the main result, the new infections, is given by the spatial transmission. I mean, with the proximity where one farm to the other is more important than the animal movements. And this is a highly expected result because this area had a higher density of cattle farms. On the other hand, in the swine populations, Given the pyramidal structure of this production chain, where we have growing chains or nursery that send animals to finishers, and these finishers send finally animals to the slaughterhouses, this creates a large network where the animal movements, they are more important than the spatial transmission. They are found that they are connected more, given by the animal movements, by the space. And this is why we found here. And also for the small ruminants, we have a similar phase to the cattle simulations, but with a lower number of infected animals. So here we're going to talk about the future steps. Right now, I'm working on the code to modeling control actions based on the spatial buffer area of 3 kilometers, 7 kilometers. And also we are considering animal band, considering staying still of 5 days, 10 days, or 50 days. We are working on this right now. We are making the simulations. Also, we are considering the contact trace by animals across the movement network. And also we are considering test and cool in different surveillance areas. We are working this. We want to show in the future more results about that, but these simulations takes a lot of time. It's highly computational, expensive. So that was the reason we don't have so much results right now, but we're gonna be later with more results than this. So thank you so much. I'll remember, my name is Nicolas Cespedes Cardenas. This is my email. This is the website of the laboratory. And I want to say thank you so much for Fundesa for supporting this work, and see you later. And thank you so much.
Hello, everybody. Welcome to my presentation. I'm Victoria Iriarte. I'm a veterinarian of the epidemiology unit of the Ministry of Livestock, Agriculture and Fisheries of Uruguay. I'm a PhD student as well in Wageningen University and Veterinary School of Uruguay. My project is funded by National Research Innovation Agency. My promoter is Professor Mar de Young, and my supervisors are Jose Gonzalez and Andrés Gil. Before getting into the main point of my presentation, I want to tell you that my PhD project is about foot-and-mouth disease transmission and effective emergency response in Uruguay. During my PhD, I am planning to do an epidemiological study of the 2001 epidemic in Uruguay. I will quantify transmission. We want to assess the effectiveness of the measures taken during the epidemic, using kernel models, and finally we want to use those models to the current livestock population in Uruguay to predict the impact of future epidemics and to identify effective control measures that would lead in an optimization of the emergency preparedness program. So today I want to talk with you about some results of my first chapter. I divided this presentation into 3 main points. I will start with an introduction, then I will show you some results of a model developed to estimate the date of the virus introduction. Then we are going to talk about the results of a multivariable analysis to identify risk factors during the high-risk period. And finally, I will summarize the main points we discussed. So to start, let's start with the 2001 foot-and-mouth disease epidemic in Uruguay. As I am sure you all know that in 2001, Uruguay experienced a virus introduction that led to a large epidemic. So the first outbreak was detected in April. And first, authorities sought to control the epidemic without vaccination, implementing measures such as banning of animal movement, stamping out of sick animals and in contact, first locally, then extended to a national scale. Then stamping out was stopped, an emergency ring vaccination was implemented, and later in the epidemic, the infection seems keep spreading, so a mass vaccination of the entire cattle population was adopted, and the last outbreak was in August. At the end of the epidemic, 2,057 farms were infected. So let's move to the second part of this presentation. After a first exploration of the data, we realized that there were some uncertainties regarding the date of the estimated date of onset of symptoms. So we use this model, SCIR model, trying to estimate the date of the virus introduction from the number of diseased animals. In the table, we can see the parameters we use, and we set different scenarios for dairy and beef farms. I won't get deeper into that, but basically what we did is to look at the time when 5, 15, and 40, and so on animals were diseased. And we set some general rules. For example, for dairy farms with less than 5 animals, we assume that the virus was introduced 7 days before. What I want to show you is that when we use these corrected dates in the data set, it seems that by the time the first outbreak was detected in Uruguay, more than 200 farms were infected and already spread across the country. So we can see that probably this hampered the efficacy of the control measures taken during the first stage of the epidemic. I divided the epidemic into 4 main periods: the high-risk period in red, the period where before control measures were implemented; in blue, this period when they tried to control the epidemic without vaccination; and then the first round mass vaccination and the second. But today we are going to focus on the high-risk period with 44,693 negative farms and 242 outbreaks. Let's move to the last part of the presentation. The goal of this study was to identify risk factors associated with foot-and-mouth disease virus introduction and infection during the high-risk period. First, we perform an univariable analysis. In this table, we can see the results, but I won't get deeper into that because I want to focus on the results of the multivariable analysis. But I want to show you which variables we explore. So first, herd density, cropping. Those farms that have a mixed production system combining livestock with cropping are classified as yes. The distance from Argentina border measured as stripes, each 50 kilometers, the distance from main routes, animal movements, farm size, species kept, only cattle, mixed, on sheep, the presence of pig, the cattle young stock proportion, and finally, if the farm was a dairy farm or not. So we can see here the results of the multivariable logistic regression. We have the result, the odds ratio for each variable adjusted for the other variables included in the model. First, we can see that the odds of being infected decreases when the distance from Argentina increases, and the odds of being infected for those farms that had cropping as an activity are 2.68 times the odds of being infected for those that don't have cropping as an activity. And I can relate these two variables with foot-and-mouth disease virus introduction, because the main hypothesis stated in Uruguay during the epidemic was that the virus was introduced from Argentina by fomite, because the area, the main area affected involves an intense cross movement of people, hunters, people related with the agriculture sector, farm equipment. So I can see this cropping as an indicator of those farms which involves this intense cross movement of farm equipment, people, etc. We can see that the odds of being infected for large farms are higher than the odds of being infected for those smaller farms. Regarding the species, we can see that the odds of being infected for mixed farms were 20% less than the odds of being infected for only cattle farms. The odds of being infected for those farms with movements are higher than the odds of being infected for those farms without movement, we can see that the odds of being infected for those farms located near routes are 36% higher than the odds of being infected for those farms located more than 100 meters from main routes. And finally, we can see that the odds of being infected for dairy farms are 68% higher than the odds of being infected for non-dairy farms. And here we consider the same results, but in a probability scale, which can have a straightforward interpretation. So we can see that the probability of infection was high for those farms near the border from Argentina that have cropping activity with animal movements, that keep only cattle, and that were dairy farms. So let me summarize the main points we talked today. First, that more than 200 farms were infected and already spread across the country by the time the first outbreak was detected. That highlights the importance of early detection. Second, we identified some risk factors during the high-risk period. Proximity to Argentina and cropping related with foot-and-mouth disease virus introduction, and large farms, proximity to main routes, cattle and dairy farms were associated with high risk of infection. And finally, that the movement of infected animals before the restrictions were implemented seems to have played an important role in the spread of the disease, like in other major epidemics. That brings me to the end of my presentation. If you have any questions, I would be happy to answer them. Thank you very much.
Hi everyone, today I'm going to talk to you about use of slaughterhouse as a sentinel points for genomic surveillance of foot-and-mouth disease virus in southern Vietnam. Foot-and-mouth disease is caused by a virus, as you already know, that affect cattle, pigs, and other ruminants. The disease produce clinical signs of blisters in mouth, hoof, tongue, and other that lead to economic losses. There are 7 different serotypes of the virus, and many viral lineages that circulate in the world have emerged from Asia. And in order to eradicate the disease from the world, it's really important to know what is the FMD situation in Asia. Here I'm showing the livestock density map of Southeast Asia, taken from FAO. And green color arrow shows cattle and buffalo movement occurring from borders of India to Southeast Asia. And the red color arrow shows pig movement occurring to and from Malaysia to China. And Vietnam is located at the center of this animal movement, and knowing the FMD situation in Vietnam would provide us information about the FMD situation in Southeast Asia. In Vietnam, livestock economy is based on pig production and also on cattle and buffalo production. FMD is endemic to Vietnam, and currently serotype O and A are circulating in the country. High-risk areas for FMD in north and south part have been identified in a previous study conducted in 2020. And Vietnam is now in stage 3 of the progressive control pathway introduced by FAO and OIE. And to know the FMD circulation in Vietnam, we could perform surveillance. And then when it comes to surveillance, if we just look at the reported outbreak numbers, that would be looking at tip of an iceberg, whereas population level virus circulation remain largely unknown. And active surveillance is better than passive surveillance, where we could get a better idea. Active surveillance is carried out in farms and slaughterhouses. Oftentimes, surveillance at the farms is not very cost effective, and biosecurity-wise safe. Therefore, if we could get the same information from slaughterhouses, that would be a better opportunity. So the objectives of this study were to quantify whether viruses isolated from slaughterhouses are representative of circulating FMDB strains, and to evaluate the timeliness of slaughterhouse surveillance for early detection of outbreak strains. As I have already mentioned, north, central, and southern part of the country have been previously identified as FMD high-risk areas, and those areas are where our samples are mainly coming from. And for farm-based sampling, this was carried out from 2015 to 2019 as a serial cross-sectional sampling study. And 4 provinces in North Vietnam and 4 provinces in South Vietnam were included, and specifically 1,246 farms in North Vietnam and 576 farms in South Vietnam to obtain serology data and then VP1 sequences. Slaughterhouses are located in these 2 provinces in southern Vietnam, where sampling is carried out from 2017 to 19. A cross-sectional sampling that is carried out every 15 days, where we obtain number of VP1 sequences, and also we have NSP serology data. And outbreak sequences that we used in this study are coming from 38 provinces, representing different provinces, areas of the country, such as north, central, and southern part. These sequences are coming from GenBank. So the objective of this study was to quantify whether viruses isolated from slaughterhouses are representative of circulating FMDB strains. In order to do this, we created phylogenetic trees for both serotype O and A. And here I'm showing only the serotype O tree. And as you can see, these different sequences belong to 4 different lineages that were circulating in Vietnam at that time. The tip color shows where the samples have been collected, whether it's a farm, outbreak, or slaughterhouse. And then the branch color shows different region, whether it's central, north, or south part of the country where it was collected. And a similar kind of tree was created for serotype A as well, which I'm not showing here. So we used this tree to quantify whether viruses isolated from slaughterhouses are representative of circulating strains. To do that, we performed a discriminatory principal component analysis, which provided us with these different clusters. Here I'm showing clusters which had sequences greater than 10. And here I'm showing the serotype O6 clusters. And then if we look at these clusters for both serotypes, we can see that slaughterhouse surveillance detected 6 out of 7 genetic clusters that was circulating during this period. And these clusters appeared in different times. For example, cluster 1 appeared earlier before cluster 10, and that appeared before cluster 2. And if we focus on this one big cluster, cluster 2, we can clearly see that sequences from slaughterhouses that would cluster with outbreak sequences would appear earlier. And here I'm showing the same information in the map, where we can see slaughterhouse sequences 2017 in this particular cluster. And then in 2018, we can see outbreak sequences that clustered with the same slaughterhouse sequences would appear in north, central, and south part of the country, indicating that slaughterhouses could detect similar sequences to outbreak sequences before they emerge as outbreak strains. This study had several limitations. One is we also look at the serology data, NSP response, to compare farms and slaughterhouses, and those results were inconclusive. And then looking at the serotype O3, that one cluster we did not identify that was in southern province in slaughterhouses that belonged to a pig outbreak. We could not detect it in bovine slaughterhouses. And as I've shown that these genetic clusters change over time, but we do not have enough evidence to say it conclusively because of the limited sample size. And since these slaughterhouses were in southern part of the country, it is not representative of what is happening throughout the whole Vietnam. In conclusion, this study demonstrates the potential utility of slaughterhouse-based genomic surveillance, and this sort of routine genomic surveillance is good to detect establish and also emerging genetic variants, and in some cases, their detection before passive surveillance. And this kind of broader network of slaughterhouses could be employed as a part of a national surveillance strategy. And these steps would be important to support PCP's stage 2 activities, such as monitoring of circulating strains in a country. And then also in the stage 3, when a country is going toward zonal freedom, this would be a better approach for rapid detection of outbreaks. This study was a product of a tremendous amount of work from our collaborators in Vietnam. So I would like to acknowledge them, and also Plum Island Animal Disease Center, USDA, and then University of Minnesota. Thank you very much. And with that, I could take any questions.
Good morning, good afternoon, and good evening. In the next few minutes, I would like to share with you the initiative by FAO and OIE towards the secure elimination of foot and mouth disease serotype C. What we're going to cover here is the background and objectives, draft action plan for this initiative and proposed activity. Then we have a conclusion and a way forward. In the background, as you an FMD expert, you probably aware that we have not seen FMD cases with serotype C in susceptible population, perhaps since 2004. So we're talking about over 17 years with no detection of this serotype in the field. We have some evidence and some data to show that there was no serotype C cases reported in Europe since the vaccination stopped in 1990. We also have some information or evidence to show that serotype C has not been reported in Asia since 1994. Historical data, however, showed that there were some outbreaks with virus that's closely related to vaccine strains, which might indicate that perhaps the vaccine virus was not fully inactivated, or there was escape of virus from laboratory. That is shown in Europe and East Africa. In South America, there were compelling evidence through studies and so monitoring of serotype C in the continent, showing that there were no cases detected up till 1994. Then there was some incursion of serotype C in Amazon in 2004. So there were a great deal of studies done looking at data and samples from 1968 to 2016 to look for this particular serotype, and it was indicated there is no virus circulation in the population. So that's really a good evidence for us to think, okay, let's take this further and proven that the virus is perhaps extinct. So the initiative between the two organizations were to encourage and compile some evidence, body of evidence. We need to have compelling evidence in our hands to show that virus is no longer circulating, particularly the serotype, if I have to be correct on that. Then we need to conduct further investigation on some historical samples being collected over time to see these samples were not containing FMD serotype C. So another objective we need to carry on is to reduce the risk of any release of this serotype from laboratory vaccine production facility as well. Then we maintain a system of preparedness to immediately detect and respond if the virus reintroduced. So the initial activities taken on by the two organizations were to formulate a task force is under the platform of global framework of transparent animal diseases, which is all the activity between the organization were taken on. So there were a task force put together with representatives from FAO, YE, and WRL reference center. 2 meetings have been taking place in September and October, and during these 2 meetings, we were able to come up with a draft outline of the action plan, considering the next 5 years. And the action plan, that's a draft, again, to put together evidence and measure risk. We need to have compelling evidence to show that particular serotype is gone. Phase 2, after we have all this evidence, we need to reduce the risk after it's been identified and maintain preparedness. So, as I mentioned, 2 phases of the action plan. Under the first phase, it's to estimate the confidence level that this virus is gone, or this serotype is gone. And to also gather information regarding where the virus is kept, where the serotype strains are kept, in laboratory, vaccine companies, and somewhere else. After we do that first phase, we're looking at how we're going to reduce the risk based on the data from phase 1, how we're going to reduce the risk and how we maintain preparedness. All along, as we're doing this, we are going to communicate and have outreach with the stakeholders. We're all on the same page because it's a concerted effort that has to be done at the national, regional, and international levels. So in more details about the phase 1, gathering evidence and measuring risk, we will analyze data that's present in the OIE and FAO reference centers, and also other places where we can find information. And we also have questionnaires to be sent out to basically identify where the virus strains of the serotypes are kept, and maybe perhaps an inventory. Similar, this is basically similar to what we have been doing for Interpest cluster eradication, where the virus is basically gone from the susceptible population, but we do have a lot of countries, several countries keeping the virus in their laboratories for one reason or the other. However, we were able to convince, in the best case, we're able to convince country to destroy their stock or transfer it to a holding facility. This is the communication piece and outreach that we have to have with this type of initiative. Once it's all done, we're going to do a risk assessment and cost benefit to move the stage, the phase 2. In phase 2, we are going to start with having, by putting together, getting a joint advisory committee to help the two organizations in moving forward. The activities that we have proposed currently is, of course, communication and outreach to member countries, and that's the first initiative, and also particularly vaccine manufacturers and laboratory. So this is, we need to share with them our initiative and to come up with an agreement to stop holding, using the virus or working with the virus. And also establishing a holding facility where we keep the valuable strain of serotype C. And this facility, as we've done with Rinderpest, we're going to be evaluated against the international standard of biosafety and biosecurity. Then we have to scrutinize dealing or handling the live virus in any type of research initiative. So the approval will be needed from the two organizations through the support and recommendation from the Joint Advisory Committee. We're also planning to have validate non-infectious diagnostic test for country to use when they are working on an outbreak investigation. They can use that test to rule out serotype C. We need to also, for preparedness, we need to have a vaccine reserve available and also have a contingency plan that's being practiced and exercised to make sure it can be used if the virus returns. Also, we need to develop a policy to reduce the malicious use of this virus. In conclusion, just concluding what I just said, and I would like to bring to your attention that the OIE resolution 30th that came out or adopted in 2017, which basically paving the road to the initiative we're doing right now, is member to investigate suspected cases of foot-and-mouth disease and to rule out FMD serotype C, and if it does find it in an outbreak, should be reported. Assess the risk and the use of serotype C in a vaccine, and stop the use of this virulent virus in a challenge study that's usually done for validation for the efficacy of efficacy study of the vaccine, and also to halt vaccine production. This is encouraging country to do that in 2017, and also to urge reference center to participate and contribute to diagnostic and research for our surveillance activities. So I mentioned the FAO-OIE task force is already in place. I also mentioned that the phases we have are basically going to be a progressive approach, looking at what we are finding, and from there we can adjust our activities based on risk, buy-in, and also cost. So we understand this initiative, as we have done within the best poster education, is got a set of challenges. However, we have learned quite a bit from that experience, and we're able to work on it and work with countries to get them to understand the benefits and understand and buy in, and their contribution will be very aligned with the regional and international effort. So the challenges are there, competing priorities are there, but we think this is a way forward for us to perhaps look at the next, perhaps hanging fruit of maybe eliminating some of the FMD strain or FMD serotypes in the future. So I would like to acknowledge the Global Framework of Transparent Animal Disease Management Committee and the Secretariat, and also have a list of here for you to know who's on a task force, initial task force, and to also acknowledge the work done by David Payton, who actually have a good, nice publications on the history of serotype C, and for Antonello Di Naro, we actually work on analyze the data. With that, I want to thank you so much for your attention, and please let me know if you have any questions.
Right. Thank you to all the speakers for their contributions to today's session. Very interesting and covering a large diversity of different epidemiological research and other efforts worldwide. So for this discussion, we have been fielding questions that are coming through the chats as well as from one speaker to another. And Carson and I will do our best to moderate this discussion. The first question that we have is going to be for our invited speaker, Dr. Heath, on the FMD outbreaks that have been documented in South Africa. And the questions, so there was actually two that came in related to the available serology data. And the first is about post-vaccination monitoring and whether this is done, and if yes, did most vaccinated cattle have a good vaccination response? And then a related question about serology data. Can you tell us more about the level of NSP serosurveillance that is routinely undertaken in the high-risk areas of South Africa? And does these data, or do these data provide any evidence for FMD circulation in the absence of clinical disease?
Okay, appreciate that. Two very interesting questions. I'll start off with a post-vaccination surveillance. Unfortunately, post-vaccination surveillance is not a routine activity in South Africa. There are several reasons for this, mainly related to the availability of the vaccine. Unfortunately, in recent years, South Africa has struggled to secure regular supply of vaccines, which has made the application of vaccines a little bit sporadic and usually focused around outbreaks. The second contributing factor that prevents us from doing post-vaccination monitoring is that we're not sure what the strains are that's included in the BVI vaccine. So for us to do post-vaccination monitoring, we would prefer to use homologous strains, which we currently don't have access to. We did in the past try and use the strains that we have circulating in South Africa and the strains that we've identified in South Africa for diagnostic purposes. But since we cannot confirm whether these are homologous or not, the interpretation of post-vaccination data that we get is questionable. So unfortunately, it is something that we've discussed before. We would love to do, but practically at this stage, it's not being done. With regards to the NSP testing, again, South Africa takes a slightly different approach to some of the other reference labs in that we don't use NSP as a surveillance tool. We use a solid phase competition analyzer as our primary surveillance tool in the laboratory. So whenever animals move in South Africa, they need to submit samples to the laboratory for permitting. And through that, we tend to get an idea of what the surveillance is. NSP testing is done primarily for confirmation of outbreaks. So when we do have an outbreak or serological data that looks a little bit suspicious, we would then start testing using NSP. I can use two examples really in terms of answering the second part of the question in terms of whether it identifies circulation of virus in the absence of clinical disease. And firstly, that is the outbreaks that we had in 2019 in the free zone, where the animals in a feedlot were affected initially, and from there it spread a little bit more. In South Africa, we don't have a culling procedure. So although we encourage owners to cull the herds that are affected, it doesn't always happen. It's at the discretion of the owner. So we've monitored some of the affected herds over 18 months using NSP as well. And in those herds, we do see a large proportion of the animals testing positive, both on solid phase and on NSP without showing clinical disease. The second example is what we saw in Natal, similar situation there, in that animals did not present with clinical disease in most cases, not in all, but in most cases. But the percentage of positives there, both solid phase and competition ELISA were quite significant. In Natal, we tested approximately 26,000 animals over a 3 to 4 month period. The positivity rate on that was around about 10% on solid phase competition ELISA and very similar for the NSP. So the correlation between the two tests is good, but we don't use it routinely to identify circulation of viruses without clinical presentation. I hope that answers the questions satisfactorily.
Yes, and I have one follow-up. You mentioned that you believe that the epidemiology of the disease has been shifting in the last, say, decade or so. And I guess I was wondering your thoughts. Do you think it's something that's been changing with the way buffalo are managed or the way that wildlife-livestock interface is happening? Or do you think that some of the change in epidemiology might have something to do with the surveillance that's being done and the ability to detect outbreaks before they become widespread?
Yeah, unfortunately, I think it's the second option more so than the first. The epidemiology with the wildlife interface and the involvement of buffalo clearly is still a major factor in South Africa. We've had several outbreaks where we could identify contact with buffalo as the primary source of infection. So that remains important. But unfortunately, I think the ability to detect outbreaks when they occur and as soon as they occur and to control that has contributed to the change in epidemiology, where we see cattle to cattle spread becoming much more important, specifically with the incursion of the disease into the free zone. Buffalo movement into the free zone is very tightly controlled. No buffalo may move out of the control zone without being tested or moved at all in most cases. So unfortunately, we're seeing a situation where cattle to cattle spread after an outbreak has been identified or before an outbreak is identified as being a major contributing factor to this.
All right, thank you so much. There was a second question for our first speaker, Nicholas, about the modeling of FMD incursions in the southern part of Brazil. And the question is related to, I'll try to summarize, the relative importance of different types of transmission modes, because with all simulation models, you have to make some assumptions about the transmission rates by, spatial transmission versus movement transmission, et cetera. And the question was related to citing an earlier paper from a FMD outbreak in Europe about the relative importance of different transmission modes. But I was wondering a little about how you parameterized your spatial transmission and what data do you use to inform that? Because of course that affects your conclusions on the importance of spatial transmission. And is there anything we can learn about the outbreaks that Victoria presented about what happened in Uruguay in 2001 that could help inform the transmission rates that you're using for Brazil, just over the border?
Yeah. Okay. Hello. Right now it's very hard to parameterize the model. So we are using parameters for the model, but also we get data from the outbreak for 2001. in order to calculate what's the distal among the farms or what is the utilization coefficient on those farms. So we use the data from the outbreak for 2000 and 2001 to compare what is the results of the model and fit the models in order to see if the results make sense or not. With the other questions, if we have a parameter related with the movement of the beds, vomiters, and the other routes of transmission. Unfortunately, we don't have this information available right now. It's very hard to parameterize that kind of data in the model, so we don't have so much information. However, we have this information for the over 2000, 2001, so we are working to make more data and get some parameters using this information from those outbreaks that was 20 years ago, but this is the information that we have. So we are working on this right now. So right now we don't have any published information yet.
All right, thank you, Nicholas. And I had a question for Samia about the serotype C. And there was just some recent work on the differences in the epidemiology of the SAT serotypes in buffalo, and specifically how the differences amongst serotypes and the duration of immunity or duration of the carrier state, et cetera, how those differences between serotypes actually determined the ability of each serotype to persist in buffalo populations or host populations of different sizes. And so I was just wondering your opinion, is there anything that's unique about FMD serotype C in terms of its epidemiological characteristics that has made it a less fit virus and that has allowed it to potentially at least go, out of our radar for so long, if not extinct? Is there anything different about FMDC compared to other serotypes?
Thank you, Kim. This is an excellent question. I'm not sure if we have the answer for it. We know serotype C was a derivative of serotype O, like a strain that changed over time and become a different serotype. I didn't think from the data, I could be wrong. I think we need to talk to the reference centers. From the data we have, I don't know if we associated serotype C to any latent infection. I didn't think we ever came across some data to show that we have latent infection with serotype C compared to other serotypes. So it is causing clinical disease. It is as any FMD serotype, but I didn't think it is, maybe it is kind of released or disappeared or animal recovered from that serotype. That's a very, very good question, and then we'll probably need to search the data to maybe come with some evidence that this serotype had certain characteristics that made it basically extinct. And maybe that could be looking at what could be the next strain, I would say, or serotype of FMD that has a similar trend that could be extinct in the future. I think this is a very, very good question. Thank you. I don't know if I answer you because we didn't have an answer.
Yeah, and a follow-up question to that, and I think that probably a lot of people have thought similarly, is how likely do you think that that there are serotype C viruses hiding out in areas where just do not have very good surveillance outbreaks, and that it may be there, it might be locally circulating, but there's certainly countries or whole regions that we have very little data on, and so I guess I was just wondering your thoughts on how possible is it that the virus could persist there but not have really expanded to areas outside those regions where you'd be more likely to detect it.
Yeah, this is actually the work that we need to do because there was no reporting, there was no isolation of serotype C since 2004. There are some suspect cases, but it didn't come out to be serotype C. So what we are doing, we're going to look at the samples submitted perhaps to the reference laboratory and make sure it does not have serotype C. Where serotype C was reported, that's our questionnaire that's going to go to countries. Where was reported the last outbreak, for example, and we maybe need to do surveillance in that particular country, on that particular region. So this is a discovery that we're going to do in phase 1 to see really, do we have anything to suspect serotype C somewhere in Africa, and I think it was reported in Asia. South America already eliminated serotype C from their studies, and they have not reported since 2004. So we have a very good evidence this virus is gone, but we have to make sure that it's not in a susceptible population, it's not in wildlife. All of that needs to be done at this stage. Hopefully, we're hoping this will be done in the next 2 years. And then after that, we'll continue looking to see how we're gonna actually mitigate such a risk, if there is any risk.
Yeah, that's interesting. That actually blends quite nicely with our talk on slaughterhouse-based surveillance. And one could envision how, if you can't sample very effectively in some of these regions due to political instability or otherwise, that you could try to establish some sentinel points along those supply chains that are that animals are coming from some of these unstable regions, but into the supply chains in other areas of East Africa, for example, and hopefully try to capture it at those sentinel points.
Right. Yeah, true. We have to design a surveillance. We're looking at data first, and we're talking to countries. And you probably get our questionnaire as being a research institute or a reference laboratory. You're gonna get our questionnaire, and you're gonna give us some information to work from there. Yes, it's a lot of work, but I think exciting because I'm working on rinderpest post eradication activities, where we have 36 countries keeping the virus while it's all basically eradicated. And we had to work with them to know what the virus is, which lab, how many viruses do you have, many strains, can you help us to destroy it, can we sequestrate it somewhere safe? That's pretty much similar to what we've done with rinderpest. And now we only have about 6 countries, that the last 6 countries are very, we're working on them to get the virus out and destroy it if possible. So we do have quite a bit of experience in that arena. So we probably know how to deal with it, learning from rinderpest. Thank you.
All right, thanks so much.
Your talk has sparked a lot of interest, and also we have a lot of questions on for Samia. One is on the 2 phases you mentioned in your action plan, and there was a question whether the 2 phases should not be going in parallel yes, and should not be going in parallel. And also we have questions on the storage of seed virus for vaccine production, and how is the future of that, and if vaccine should not be based, or a potential C vaccine should not be based on virus like particles. Can you answer on these questions?
Yeah. Okay. So the first question is that we need to think about how we are not having overlap of the 2 phases. They will overlap, that's for sure. But we're trying to have a body of evidence, because we want to make sure we're walking in the right direction. Body of evidence to tell us the virus is not in susceptible population, it's not in the field, it's only in the laboratory. This is our first phase. And we have to do a great deal of communication with countries to get that information. So once we get that done, we're able to move into activities. I think this is what we discussed. We only had 2 meetings with the task force, so we still have a lot of thinking. If somebody has any ideas, please contact me. Maybe we can improve our thought, but this is our initial draft of how we're gonna proceed. Now, your second question about vaccine. we are thinking about the existing vaccine that we have, maybe vaccine banks, for example, in Europe, in the American side. We might have to keep those as vaccine reserves, not to use them, but keep them for emergency. I didn't know about getting a different vaccine. You know how long it takes to get a new vaccine for FMD? Many years. And I'm not sure we're going to switch into, we thought of messenger RNA vaccine would be fantastic, but it needs a lot of work to get there. Now, this is also too far for us to think about, but we already have vaccine that we can use as reserve. We limit vaccine production. That's what we're trying to do. Thank you.
All right. Well, I'd like to thank all of the speakers. Unfortunately, our time has wrapped up, but I do understand that there is a specific kind of a coffee area that if you want to continue these discussions, a lot of these conversations can continue in the coffee breaks. So if you want to follow up with any of the speakers, I hope that they'll log into that coffee room for these discussions. And thank you everyone for your engagement today. And thank you especially for our speakers for being part of this session today, as well as thank you to the organizers for making this very seamless for us.
Thank you.
Yeah, thank you for the organizer and wonderful platform for these meetings. Fantastic. Thank you.
Thank you.
Thank you so much.
Thank you. Bye-bye.